Assalamualaikum warahmatullahi wabarakatuh. We are from Group 7. We'll explain about how language is represented in the brain, neurolinguistics. First of all, I will introduce the speaker. The first speaker is me, Nur Alice. The second speaker is Nida Rohmah. The third speaker is Widi Arta and Muhammad Faza as the last speaker. Table of contents. We will explain about Chapter 1, Definition of Neurolinguistics. Chapter 2, The Biology of the Brain. Chapter 3, Question about Biology of Language. And Chapter 4, Methods in the Study of the Biology of Language. Chapter 1, The Definition of Neurolinguistics. Neurolinguistics is a field of study that combines neuroscience with language. So researchers in neurolinguistics study how our brain works when we speak, understand, and produce words. They are interested in the parts of the brain that are active when we use language, such as the parts that help us pronounce words or understand the meaning of sentences. By understanding this process, neurolinguistics can help us learn more about how our brain works when we speak and can also help people who may have problems with language due to brain injuries or disorder. One example in neurolinguistics is when researchers use brain imaging technology such as fMRI or EEG to examine brain activity when someone speaks or understands words. They can see how certain areas of the brain are active when someone speaks a certain language or when someone understands a complex sentence. Using these techniques, researchers can find out more about how the brain is involved in language process, and this could provide important insight into how language works in our mind. Chapter 2, The Biology of the Brain. Part A, The Cerebrum, the largest and most crucial part of the brain. Our large brain is divided into two parts that work together. Each part has four sections called lobes, such as the frontal, temporal, parietal, and occipital lobes. Each lobe has small hills and valleys within it. It's like the mountains and valleys on a map. Individual variation exists in the exact location and shape of gyri and sulci. In our language, gyri is punggung bukit and sulci is lembah bukit. The nomenclature often use directional terms like superior, inferior, anterior in front of, posterior behind, lateral toward the side, medial toward the center where hemisphere meet, ventral bottom, and dorsal top to describe the anatomical position within the brain. B. The cerebral cortex and its neuron. Our brain has a part called the cerebral cortex and neurons in it. The cerebral cortex is like the bark of a tree that protects the brain. Inside the cerebral cortex, there are millions of neurons that work together. These neurons are responsible for our thinking and memory. The neuron is a nerve cell, the basic unit of the nervous system responsible for transmitting, receiving, and processing electrical signal in the human body. Neurons work by sending electrical signal through long section called axon. The signal help us think and move. When this signal reach their end, they release chemical called neurotransmitter. This chemical help other neurons communicate with each other. Neurons have part called dendrites and cell bodies. These dendrites and cell body appear gray, so they are called gray matter. On the other hand, axons that are covered by a white pod-like material are called white matter. Both types of matter help our brain function properly. You can see in the picture. For example, when a person learns new word, the neurons in the brain are hard at work. The dendrites in each neuron will retrieve signal from other neurons, process information about the new word, and then transmit the information to the rest of the body through its axon. During this process, neurotransmitter in the brain also help transmit signal and information from one neuron to other, helping the brain to understand and remember the new word.
See cytoarchitectonics, the distribution of neurons in the cortex. The cerebral cortex consists of various types of neurons distributed across different layers, allowing us to distinguish brain regions based on difference in cytoarchitecture. The fundamental distinction is between the neocortex with 6 layers of nerve cells, found in nearly the entire cerebrum, and older forms of cortex, which have fewer layers and are located in specific regions of the medial temporal lobe. Finer-grained cytoarchitectonic differences in various cortical regions, consistently found in different individuals' brains, can be identified. The cerebellum, subcortical structures, and networks brain. Most studies on the neural basis of higher cognitive functions, particularly language, have traditionally concentrated on the cortical regions of the cerebrum, particularly the lateral neocortical areas. However, it has become increasingly evident that other brain structures also have a role in language processing. The cerebellum, previously associated primarily with motor functions, has accumulated evidence linking it to language, memory, emotion, and other cognitive domains. Additionally, there are subcortical structures deep within the cerebrum, like the basal ganglia and hippocampus, which were initially linked to movement and memory but are now understood to be significant in language processing. It's crucial to recognize that no single part of the brain functions in isolation. Instead, the brain operates an interconnected network of structures that collaborate to solve problems, emphasizing the importance of understanding the entire network and how each component contributes to specific functions. Chapter 3, Question about the biology of language. A. Biological substrates. What are the biological bases of language? Research on the biological basis of language involves anatomical structures, cytoarchitectonic regions, brain networks, neuron types, neurotransmitter, hormones, genes, and other biological substrates. This is essential for understanding the biological foundations of language and how it affects language processing. This research also provides insights into the structure of language. While much of the focus has been on brain anatomy, we can also examine the types of neurons, neurotransmitter, and hormones involved in language. Genetics of language has also become a significant focus, with studies seeking correlations between genes and language abilities. All of this help us understand how biological components work within language and contribute to our understanding of language.
B. What does the movie of brain activity during language use look like? We know that connecting language function to specific brain structures, cells, molecules, or genes are important. However, we also need to understand how these biological components are utilized during the actual production or comprehension of language. This spatiotemporal dynamic of language processing involves not only anatomic aspect of language but also real-time temporal interaction, such as which brain structure are activated and in what sequence. Essentially, understanding the Spatiotemporal dynamic of language processing is like watching a movie of brain activity over time. For example, studies have started to unveil the sequence of brain activity when naming pictures, processing syntax, and or forming English past tense. In a broader sense, the biological temporal dynamics of language refer to the real-time functional biology of language processing, encompassing the real-time interplay not only of the brain structure involved in language but also the cellular, molecular, and even genetic aspect of this domain. Next is chapter 4, Method in Study of the Biology of Language. A. Deletion method is used to understand the relation between brain damage and the loss of cognitive function. Consistent damage to specific brain structure resulting in the loss of function indicates the importance of the structure for this function. Although useful, this method has limitations, such as the difficulty of controlling the location and timing of lesions. While still in use, the method can become complex when applied to children or individuals with developmental brain disorders, as children's brains are highly plastic and can compensate for damage. In summary, the lesson method have a way to study how brain damage affect to cognitive function. When specific brain areas are consistently damaged and function are lost. But this method have limitations like difficulty in controlling the location and timing of brain damage. B. Hemodynamic neuroimaging. Recent technological advances now enable the imaging of brain activity during cognitive tasks in both children and adults, with or without brain and behavioral impairments. Functional magnetic resonance imaging, or fMRI, is the most widely used method, leveraging changes in brain blood flow to map cognitive activity. This method provides high spatial resolution, allowing for precise localization of brain activity. However, it cannot measure real-time processing change, necessitating other techniques with high temporal resolution such as ERPS or MEG. So, hemodynamical neuroimaging is a method that measures change in a blood flow in the brain to understand brain activity. You can see the picture. It is how this method implemented. See event related potential, or ERPS, are scalp recorded electrical brain potential that reflect cognitive process related to language comprehension. These potential are recorded in real time as subject respond is stimulus events, like words or sound, providing high temporal resolution. However, ERPS have limitation in spatial resolution and difficulty in pinpointing the exact brain structure responsible for the recorded potential. They primarily capture electrical activity from specific cortical neuron and are mainly used for language perception rather than production due to vocalization related noise. I'll explain how the method works. When the brain processing information, it generates tiny electrical signals. ERPS allows researchers to detect and analyze the signal in response to particular stimuli, helping us to understand cognitive processes like attention, memory, and language.
Method in the study of the biology of language. Point D. Magneton polycal magneto magnetoponcephalography (MEG) is a technique that detects weak magnetic field produced by brain electronical by brain electronical current by using squids. MEG provide millisecond level temporal resolution and better scores location than uh, ERPS by integrating the locate activity with MRI image research can identify likely brain structure responsible for the activity. MAG complement ERPS by capturing different brain neuron activity. And point A, direct brain recording and stimulation are employed in epilepsy patients to locate the scores of epileptic seizures, electrocode guard of the cortex surface and find probes. With electrocode interest deeper into the brain, detect signal from a few hundred cell, aiding in score location. This technique provide excellent span, excellent spatial and temporal resolution, but might not generalize to normal brain due to epilepsy-related neural abnormalities. Additionally, they can only measure activity near electronic codes necessary complementary method like fMRI for comprehensive brain mapping. And last method in the study of the biology of language point F, transcranial magnetic stimulation TMS, in a non-invasive method using rapidly charging for magnetic field to selectivity stimulate or inhibit specific brain region. It can temporarily disturb function in healthy individual for research purpose. While it offers insight into structure function relationship, TMS has limitations such as its shallow reach in critical region, short-term effect, and potential safety concern, especially in case of repeat application RTMS. Okay, thank you.
Thank you for watching and paying attention. If you have any question, please ask in the comment section below. Assalamualaikum warahmatullahi wabarakatuh.